So here's the final video for the genetics unit. This one is called gene mapping. Gene mapping is also known as gene sequencing, and I'll kind of explain as we go along the difference between the two. But essentially the idea behind gene mapping is we're going to try and figure out, looking at a chromosome, where genes actually fall on the chromosome. So to start, we are going to talk about something called gene linkage. So genes that are found on the same chromosomes are known as linked. So if you look at all the different examples we have here, okay, you'll notice that these two sister chromatids share the same genes on either side. And all the genes on one chromosome, A, B, C, are considered linked. Linked genes are usually inherited together. So that's why expressions of different eye colors, or the way your hair is a certain color, pigmentation of skin, it's why we can have a variety, is because linked genes are passed through inheritance, but they stay close together. So we see this passing on of traits with variation. So during meiosis, genes will not actually separate unless we see some crossing over. So you'll see in this picture that the process of crossing over, which happens in prophase 1 of meiosis 1, the middle picture shows some crossing over between the two chromosomes. You'll notice at the end that the two chromosomes have the crossing over occurring, and some genes that are a little bit now mixed up. So chromosome 13, first copy. So here we go. Chromosome 13, second copy. There you go. When separated during meiosis, one cell gets ABCD and the other cell gets ABCD. So what you're seeing at here is the chromosome 13 and chromosome 13, both having all the genes there. But when they separate down into meiosis 2 and down into telophase, one cell is going to have ABCD lowercase, and the other cell is going to have ABC uppercase. But if in prophase 1 crossing over occurs, okay, we're going to get what we call recombinant DNA. So we're going to pretend that right here, crossing over occurred. So it was now the switching of this part of the chromosome with this part of the chromosome. So now we have what we call recombinant DNA, or rDNA, okay? We talked about it back in chapter 18, so hopefully you may remember us talking about that. So what is the point of gene mapping? Well, the idea behind gene mapping is we want to know a linear distance of genes on chromosomes. These are derived by crossing over frequency. So how common is it for something to cross over? Distance along the chromosome affects crossing over frequencies. So further apart the genes are, the higher the crossover rate. Closer the genes are, the less likely they are to actually separate, so it has a lower crossing over rate. Crossing over frequencies are expressed in what we call map units. So 1% crossing over chance equals 1% map unit. 50% equals 50 map units. So if we take a look at this chromosome listed here, okay, you'll notice that wild type AA and mutant type AA, these are genes. All of these are different genes, okay? All of these show a number along them. That's because that's how far of a map unit they are away from each other. So 0 to 104.5. A gene that is very close to each other, like these ones right here, 0 and 13, are less likely to see crossing over between them because they're so close together. You're going to see crossing over happening in an area where there's a large gap, such as here or here. The larger the gap along where genes are spread out, the higher chance they have of a higher crossing over frequency. So crossing over frequencies equal map distance. And this is important because when we map the genes, it's going to be very, very key that we know how far they are from each other. So the further apart they are, the more likely they are to cross over and separate. The less likely to be inherited together, and genes that are close together are less likely to cross over, therefore will be inherited together. So again, these pictures are just showing you a pair of chromosomes crossing over occurring, so the recombination, and now having recombinant DNA, okay? The further the genes are apart, the more likely they will separate. So big A and big C over here are quite a distance apart. Keeping them together during the process of crossing over is very unlikely because they're too far apart. And that's what we see here. A and B are fairly close in state, but C recombination occurred here. So how do we do this in the sense of map units? We're going to play a little game called Let's Figure Out How Far Your Friends Live Away from Each Other. Okay, so here we have Arnold, Beth, Carlos, and Dina. or Deanna. Deanna? I don't know. Let's just go with Deanna. All right, so they all live along the same street. We want to know how far away they actually live from each other. So we're given some information, and we also want to put their houses in order so we know which each house is that they live in. So here's the information. Arnold lived 12 doors away from Beth. Carlos lives 11 doors away from Deanna. Beth lived 3 doors away from Carlos, and Arnold lives 4 doors away from Deanna and 15 doors away from Carlos. I know what you're thinking. This sounds like a riddle that you would get at the end of the social studies tests. It's not, okay? Instead, what this is just a map and how to actually figure out who lives where. First thing we need to do is we need to find out what is the furthest distance. So we start by drawing a linear map. On each end, we're going to have a 0, and on this end, we're going to have another number. What we need to do is we need to tentatively decide who lives the furthest away from each other. So when I read this over, Arnold lived 12 doors away from Beth. That's pretty far. But Arnold also lives 15 doors away from Carlos. So looking at my numbers, 15 is the furthest number away. So it's going to go 0 to 15 tentatively. We're going to make sure this works, and I'll show you how to check it. So we would say Arnold lives here and Carlos lives there because they live 15 doors away from each other. You always want to find the biggest distance first, then go into the center and start fiddling around with others. So what I want to do is I now need to find someone who lives a distance away from Arnold and a distance away from Carlos that I can compare. So it says here that Carlos lives 11 doors away from Deanna and Arnold lives 4 doors away from Deanna. Both living away from Deanna, I have their distance. You'll also notice their distance adds up to the number that is our total. So 11 plus 4 is 15. So I know that they're going to fall somewhere in this map. So Carlos lives quite a distance away from Deanna. So rather than putting her near Carlos, she's going to be somewhere down over here, which makes sense because she only lives 4 doors away from Deanna. Oh, I did this in a different order. Sorry. So we're going to put Deanna somewhere along here. Okay. We also know that Arnold only lives 12 doors away from Beth. So he lives 12 doors away. Beth goes somewhere along here. And that's what you see here. So Arnold lives 12 doors away from Beth. We know that leaves 4 doors away from Deanna. So now all we have to do is fill in the rest of the blanks. Do we have everybody living in the correct distance? We do know Carlos lives 11 doors away because he's at 15. Here's 4. 15 minus 4 is 11. That's 11 map units. And we also know that Beth lived 3 doors away from Carlos, which is the distance between here. So we have now successfully figured out where everybody lives and figured out the distance in between. So let's take it and look at it as if it was an actual crossing over frequency. So here I have the following crossover frequency data from Drosophila. We have bar-shaped eyes, carnation eyes, fused wings, and scalloped wings. They're all on the same chromosome, and they are this much percentage away from each other. Therefore, they are that much map units. So 2.5% would be 2.5 map units. So again, we're going to start with 0 and what we think is our largest number, which is 11. Again, sometimes it ends up not working out perfectly and you have stuff outside, but that is very rare. Okay. Next, what we need to do is we need to say, okay, I figured out that my furthest one is the carnation eyes and the scalloped wings. Okay, so carnation is 0, scalloped is 11. I now need to start plotting everything in between. So cross that off my list. I'm now going to go with the halfway point. Why? Because it makes sense. If I know that it's 11 all the way across and there's a 5.5, that's a halfway point. I'm going to plot that right in the center and get rid of it right away. So that represents the bar-shaped eyes. Why? Because it is exactly 5.5% away from C. Therefore, if it's 5.5% away from C, it's also 5.5% away from S. So there it is. I'm going to go and I'm going to look at the one in between. I know the distance from B to FV, and I also know the distance from C to FV, and I notice that they both equal 5.5%. So I know it has to fall in between C and B, simply because this adds up to 5.5%, and FV is in between these two. So that's where it falls. So again, knock two more off. I'm going to look at my very last one. My last one says that FV is 8% away from S. So I just want to make sure that's true. I plotted this at 3%, that 3 here. Okay, and 3 to 11 is 8. And that means we are good. So here's one more. We're going to construct a chromosome map using the crossover frequencies. I'm going to put my extremes down, my Z and my W. Why? Because they're the furthest ones away from each other. Now I'm going to start trying to plot things from here. Okay, so I'm going to start with, here's my first one being Y. The reason I'm choosing Y is because Y is only one map unit away from Z, so it makes sense that it would go right here. From here, I will notice that Y to W is 7. So if you look here, Y to W is 7. I've already plotted that. It's already happening. 8 minus 1 is 7. Next, I'm going to plot out X. Why? Because it's the only one we are currently missing. So X is 2 away from Y, but it is also 7 away from W. So again, the distance here is 7, or sorry, it's only 5 away, is 7. So I want to put it here, so I'm 2 away from Y, but 5 away from W. And I plot that at there. Perfect. So ladies and gentlemen, there you have it. That is how you gene map. Hopefully it made sense, and you can do all the practice questions in your notes. Have a great evening, and I'll see you on Monday.